Post-translational events. A polypeptide chain is synthesized by a complex process called translation in which the assembly of amino acids in a particular sequence is dictated by messenger RNA, mRNA. We describe how a cell promotes the proper folding of a nascent polypeptide chain and, in many cases, modifies residues or cleaves the polypeptide backbone to generate the final protein. In addition, the cell has error-checking processes that eliminate incorrectly synthesized or folded proteins. Incorrectly folded proteins usually lack biological activity and, in some cases, may actually be associated with disease. Protein misfolding is suppressed by two distinct mechanisms. First, cells have systems that reduce the chances for misfolded proteins to form. Second, any misfolded proteins that do form, as well as cytosolic proteins no longer needed by a cell, are degraded by a specialized cellular garbage disposal system. The information for protein folding is encoded in the sequence. Any polypeptide chain containing N residues could, in principle, fold into 8 N conformations. This value is based on the fact that only 8 bond angles are stereochemically allowed in the polypeptide backbone. In general, however, all molecules of any protein species adopt a single conformation, called the native state. For the vast majority of proteins, the native state is the most stably folded form of the molecule. What guides proteins to their native folded state? The answer to this question initially came from in vitro studies on protein refolding. Thermal energy from heat, extremes of pH that alter the charges on amino acid side chains, and chemicals such as urea or guanidine hydrochloride at concentrations of 6 to 8 m can disrupt the weak non-covalent interactions that stabilize the native conformation of a protein. The denaturation resulting from such treatment causes a protein to lose both its native conformation and its biological activity. Many proteins that are completely unfolded in 8-M-urea and beta-mercaptoethanol, which reduces disulfide bonds, spontaneously renature, refold into their native states when the denaturing reagents are removed by dialysis. Because no cofactors or other proteins are required, in vitro protein folding is a self-directed process. In other words, sufficient information must be contained in the protein's primary sequence to direct correct refolding. The observed similarity in the folded, three-dimensional structures of proteins with similar amino acid sequences provided other evidence that the primary sequence also determines protein folding in vivo. Folding of proteins in vivo is promoted by chaperones. Although protein folding occurs in vitro, only a minority of unfolded molecules undergo complete folding into the native conformation within a few minutes. Clearly, cells require a faster, more efficient mechanism for folding proteins into the correct shapes. Otherwise, cells would waste much energy in the synthesis of non-functional proteins and in the degradation of misfolded or unfolded proteins. Indeed, more than 95% of the proteins present within cells have been shown to be in their native conformation, despite high protein concentrations 200–300 mg per milliliter, which favor the precipitation of proteins in vitro. The explanation for the cell's remarkable efficiency in promoting protein folding probably lies in chaperones, a class of proteins found in all organisms from bacteria to humans. Chaperones are located in every cellular compartment, bind a wide range of proteins, and function in the general protein folding mechanism of cells. Two general families of chaperones are recognized. Molecular chaperones, which bind and stabilize unfolded or partly folded proteins, thereby preventing these proteins from aggregating and being degraded. Chaperonins, which directly facilitate the folding of proteins. Molecular chaperones consist of HSP70 and its homologs, HSP70 in the cytosol and mitochondrial matrix, BIP in the endoplasmic reticulum, and DNAK in bacteria. First identified by their rapid appearance after a cell has been stressed by heat shock, HSP70 and its homologs are the major chaperones in all organisms. HSP70 is a constitutively expressed homolog of HSP70. When bound to ATP, HSP70-like proteins assume an open form in which an exposed hydrophobic pocket transiently binds to exposed hydrophobic regions of the unfolded target protein. Hydrolysis of the bound ATP causes molecular chaperones to assume a closed form in which a target protein can undergo folding. The exchange of ATP for ADP releases the target protein, figure A, top. This cycle is speeded by the co-chaperone HSP40 in eukaryotes. In bacteria, an additional protein called GARP also interacts with DNAK, promoting the exchange of ATP for the bacterial co-chaperone DNAJ and possibly its dissociation. Molecular chaperones are thought to bind all nascent polypeptide chains as they are being. Institute for Advanced Studies, Jodhpur 39. Synthesized on ribosomes. In bacteria, 85% of the proteins are released from their chaperones and proceed to fold normally. An even higher percentage of proteins in eukaryotes follow this pathway. The proper folding of a large variety of newly synthesized or translocated proteins also requires the assistance of chaperonins. These huge cylindrical macromolecular assemblies are formed from two rings of oligomers. The eukaryotic chaperonin trick consists of 8 subunits per ring. In the bacterial, mitochondrial, and chloroplast chaperonin, known as GRO-EL, each ring contains 7 identical subunits, figure B. The GRO-EL folding mechanism, which is better understood than trick-mediated folding, serves as a general model, figure A, bottom. In bacteria, a partly folded or misfolded polypeptide is inserted into the cavity of GRO-EL, where it binds to the inner wall and folds into its native conformation. In an ATP-dependent step, GRO-EL undergoes a conformational change and releases a folded protein, a process assisted by co-chaperonin, GRO-ES, which caps the ends of GRO-EL. Many proteins undergo chemical modification of amino acid residues. Nearly every protein in a cell is chemically modified after its synthesis on a ribosome. Such modifications, which may alter the activity, lifespan, or cellular location of proteins, entail the linkage of a chemical group to the free NH2 or COOH group at either end of a protein or to a reactive side chain group in an internal residue. Although cells use the 20 amino acids shown in figure 2 to synthesize proteins, analysis of cellular proteins reveals that they contain upward of 100 different amino acids. Chemical modifications after synthesis account for this difference. Acetylation, the addition of an acetyl group, CH3CO, to the amino group of the N-terminal residue, is the most common form of chemical modification, affecting an estimated 80% of all proteins. This modification may play an important role in controlling the lifespan of proteins within cells because non-acetylated proteins are rapidly degraded by intracellular proteases. Residues that are near the termini of some membrane proteins are chemically modified by the addition of long lipid-like groups. The attachment of these hydrophobic tails, which function to anchor proteins to the lipid bilayer, constitutes one way that cells localize certain proteins to membranes. Acetyl groups and a variety of other chemical groups can also be added to specific internal residues in proteins, figure 2. An important modification is the phosphorylation of serine, threonine, tyrosine, and histidine residues. We will encounter numerous examples of proteins whose activity is regulated by reversible phosphorylation and dephosphorylation. The side chains of asparagine, serine, and threonine are sites for glycosylation, the attachment of linear and branched carbohydrate chains. Many secreted proteins and membrane proteins contain glycosylated residues. Other post-translational modifications found in selected proteins include the hydroxylation of proline and lysine residues in collagen, the methylation of histidine residues in membrane receptors, and the gamma carboxylation of glutamate in prothrombin, an essential blood clotting factor. A special modification, discussed shortly, marks cytosolic proteins for degradation. Institute for Advanced Studies, Jodhpur 40. Peptide segments of some proteins are removed after synthesis. After their synthesis, some proteins undergo irreversible changes that do not entail changes in individual amino acid residues. This type of post-translational alteration is sometimes called processing. The most common form is enzymatic cleavage of a backbone peptide bond by proteases, resulting in the removal of residues from a C or N-terminus of a polypeptide chain. Proteolytic cleavage is a common mechanism for activating enzymes that function in blood coagulation, digestion, and programmed cell death. Proteolysis also generates active peptide hormones, such as EGF and insulin, from larger precursor polypeptides. An unusual and rare type of processing, termed protein self-splicing, takes place in bacteria and some eukaryotes. This process is analogous to editing film. An internal segment of a polypeptide is removed and the ends of the polypeptide are rejoined. Unlike proteolytic processing, protein self-splicing is an autocatalytic process, which proceeds by itself without the participation of enzymes. The excised peptide appears to eliminate itself from the protein by a mechanism similar to that used in the processing of some RNA molecules. In vertebrate cells, the processing of some proteins includes self-cleavage, but the subsequent ligation step is absent. One such protein is hedgehog, a membrane-bound signaling molecule that is critical to a number of developmental processes. Ubiquitin marks cytosolic proteins for degradation in proteasomes. In addition to chemical modifications in processing, the activity of a cellular protein depends on the amount present, which reflects the balance between its rate of synthesis and rate of degradation in a cell. Here we examine protein degradation, focusing on the major pathways for degrading cytosolic proteins. The lifespan of intracellular proteins varies from as short as a few minutes for meiotic cyclins, which help regulate passage through mitosis, to as long as the age of an organism for proteins in the lens of the eye. Eukaryotic cells have several intracellular proteolytic pathways for degrading misfolded or denatured proteins, normal proteins whose concentration must be decreased, and extracellular proteins taken up by the cell. One major intracellular pathway is degradation by enzymes within lysosomes, membrane-limited organelles whose acidic interior is filled with hydrolytic enzymes. Lysosomal degradation is directed primarily toward extracellular proteins taken up by the cell and aged or defective organelles of the cell. Distinct from the lysosomal pathway are cytosolic mechanisms for degrading proteins. Chief among these mechanisms is a pathway that includes the chemical modification of a lysine side chain by the addition of ubiquitin, a 76-residue polypeptide, followed by degradation of the ubiquitin-tagged protein by a specialized proteolytic machine. Ubiquitination is a three-step process, figure 3a. Activation of ubiquitin-activating enzyme E1 by the addition of a ubiquitin molecule, a reaction that requires ATP. Transfer of this ubiquitin molecule to a cysteine residue in ubiquitin-conjugating enzyme E2. Formation of a peptide bond between the ubiquitin molecule bound to E2 and a lysine residue in the target protein, a reaction catalyzed by ubiquitin ligase E3. This process is repeated many times, with each subsequent ubiquitin molecule being added to the preceding one. The resulting polyubiquitin chain is recognized by a proteasome, another of the cell's molecular machines, figure 3b. The numerous proteasomes dispersed throughout the cell cytosol proteolytically cleave ubiquitin-tagged proteins in an ATP-dependent process that yields short, 7 to 8 residue peptides and intact ubiquitin molecules. Cellular proteins degraded by the ubiquitin-mediated pathway fall into one of two general categories: 1. Native cytosolic proteins whose lifespans are tightly controlled, and 2. Proteins that become misfolded in the course of their synthesis in the endoplasmic reticulum (ER). Both contain sequences recognized by the ubiquitinating enzyme complex. The cyclins, for example, are cytosolic proteins whose amounts are tightly controlled throughout the cell cycle. These proteins contain the internal sequence argXX-lubli-X-YLBli-AS, ASN-X can be any amino acid, which is recognized by specific ubiquitinating enzyme complexes. At a specific time in the cell cycle, each cyclin is phosphorylated by a cyclin kinase. This phosphorylation is thought to cause a conformational change that exposes the recognition sequence to the ubiquitinating enzymes, leading to degradation of the tagged cyclin. Similarly, the misfolding of proteins in the endoplasmic reticulum exposes hydrophobic sequences normally buried within the folded protein. Such proteins are transported to the cytosol, where ubiquitinating enzymes recognize the exposed hydrophobic sequences. The immune system also makes use of the ubiquitin-mediated pathway in the response to altered self cells, particularly virus-infected cells. Viral proteins within the cytosol of infected cells are ubiquitinated and then degraded in proteasomes specially designed for this role. The resulting antigenic peptides are transported to the endoplasmic reticulum, where they bind to class 1 major histocompatibility complex (MHC) molecules within the air membrane. Subsequently, the peptide MHC complex is moved to the cell membrane where the antigenic peptides can be recognized by cytotoxic T lymphocytes, which mediate the destruction of the infected cells. Digestive proteases degrade dietary proteins. The major extracellular pathway for protein degradation is the system of digestive proteases that breaks down ingested proteins into peptides and amino acids in the intestinal tract. 3 classes of proteases function in digestion. Endoproteases attack selected peptide bonds within a polypeptide chain. The principal endoproteases are pepsin, which preferentially cleaves the backbone adjacent to phenylalanine and leucine residues, and trypsin and chymotrypsin, which cleave the backbone adjacent to basic and aromatic residues. Exopeptidases sequentially remove residues from the N-terminus aminopeptidases or C-terminus carboxypeptidases of a protein. Peptidases split oligopeptides containing as many as about 20 amino acids into D and tripeptides and individual amino acids. These small molecules are then transported across the intestinal lining into the bloodstream. To protect a cell from degrading itself, endoproteases and carboxypeptidases are synthesized and secreted as inactive forms: zymogens pepsin by chief cells in the lining of the stomach, the others by pancreatic cells. Proteolytic cleavage of the zymogens within the gastric or intestinal lumen yields the active enzymes. Intestinal epithelial cells produce aminopeptidases and the D and tripeptidases. D1. Regulation of gene expression in prokaryotes, Operon concept. Jacob and Monod proposed the Operon model in 1961 for the coordinate regulation of transcription of genes involved in specific metabolic pathways. The operon is a unit of gene expression and regulation which typically includes the structural genes, any gene other than a regulator, for enzymes involved in a specific biosynthetic pathway whose expression is coordinately controlled. Control elements such as an operator sequence, which is a DNA sequence that regulates transcription of the structural genes. Regulator genes whose products recognize the control elements, for example a repressor which binds to and regulates an operator sequence. 1.1 The lactose operon. Escherichia coli can use lactose as a source of carbon. The enzymes required for the use of lactose as a carbon source are only synthesized when lactose is available as the sole carbon source. The lactose operon, or LAC operon, consists of three structural genes: LACH, which codes for beta-galactosidase, an enzyme responsible for hydrolysis of lactose to galactose and glucose; LACE, which encodes the galactoside permease which is responsible for lactose transport across the bacterial cell wall; and LACA, which encodes the thiogalactoside transacetylase. The three structural genes are encoded in a single transcription unit LAC-ZYA, which has a single promoter, PLAC. This organization means that the three lactose operon structural proteins are expressed together as a polycistronic mRNA containing more than one coding region under the same regulatory control. The LAC-ZYA transcription unit contains an operator site OLAC which is positioned between bases -5 and +2 at the 5' end of the PLAC promoter region. This site binds a protein called the LAC repressor which is a potent inhibitor of transcription when it is bound to the operator. The LAC repressor is encoded by a separate regulatory gene LACI which is also a part of the lactose operon. LACI is situated just upstream from PLAC. The LAC repressor. The LACI gene encodes the LAC repressor, which is active as a tetramer of identical subunits. It has a very strong affinity for the LAC operator binding site, while it also has a generally high affinity for DNA. The LAC operator site consists of 28 BP which is palindromic. A palindrome has the same DNA sequence when one strand is read left to right in a 5' to 3' direction and the complementary strand is read right to left in a 5' to 3' direction. This inverted repeat symmetry of the operator matches the inherent symmetry of the LAC repressor which is made up of 4 identical subunits. In the absence of lactose, the repressor occupies the operator binding site. It seems that both the LAC repressor and the RNA polymerase can bind simultaneously to the LAC promoter and operator sites. The LAC repressor actually increases the binding of the polymerase to the LAC promoter by 2 orders of magnitude. Induction. It means that when glucose is absent and lactose is present, LAC repressors cannot bind to the OLAC operator DNA sequence. RNA polymerase is also likely to be bound to the plaque promoter sequence. In the presence of lactose, permease allows its uptake, and beta-galactosidase catalyzes the conversion of some lactose to allolactose. Allolactose acts as an inducer and binds to the LAC repressor. This causes a change in the conformation of the repressor tetramer, reducing its affinity for the LAC operator. The removal of LAC repressor from the operator site allows the polymerase, which is already sited at the adjacent promoter, to rapidly begin transcription of the LAC-ZYA genes. Thus, the addition of lactose, or a synthetic inducer such as isopropyl beta-thiogalactopiranocide (IPTG), very rapidly stimulates transcription of the lactose operon structural genes. The subsequent removal of the inducer leads to an almost immediate inhibition of this induced transcription, since the free LAC repressor rapidly reoccupies the operator site and the LAC-ZYA RNA transcript is extremely unstable. CAMP receptor protein. The plaque promoter is not a strong promoter. Plaque and related promoters do not have strong -35 sequences and some even have weak -10 consensus sequences. For high-level transcription, they require the activity of a specific activator protein called CAMP receptor protein, CRP. CRP may also be called catabolite activator protein or CAP. When glucose is present, E. coli does not require alternative carbon sources such as lactose. CRP, which exists as a dimer which cannot bind to DNA on its own, nor regulate late transcription. Glucose reduces the level of CAMP in the cell. When glucose is absent, the levels of CAMP in E. coli increase and CRP binds to CAMP, forming CRP-AMP complex which binds to the lactose operon promoter plaque just upstream from a site for RNA polymerase. CRP binding induces a 90 degrees bend in DNA and this is believed to enhance RNA polymerase binding to the promoter, enhancing transcription by 50-fold. The CRP binding site is an inverted repeat and may be adjacent to the promoter, as in the lactose operon, may lie within the promoter itself, or may be much further upstream from the promoter. Differences in the CRP binding sites of the promoters of different catabolic operons may mediate different levels of response of these operons to CAMP in vivo. 1.2 TRP operon. The TRP operon encodes 5 structural genes whose activity is required for tryptophan synthesis. The operon encodes a single transcription unit which produces a 7 kilobits transcript which is synthesized downstream from the TRP promoter and TRP operator sites PTRP and OTRP. Like many of the operons involved in amino acid biosynthesis, the TRP operon has evolved systems for coordinated expression of these genes when the product of the biosynthetic pathway tryptophan is in short supply in the cell. As with the LAC operon, the RNA product of this transcription unit is very unstable, enabling bacteria to respond rapidly to changing needs for tryptophan. The TRP repressor, a gene product of the separate TRPR operon, the TRP repressor, specifically interacts with the operator site of the TRP operon. The symmetrical operator sequence, which forms the TRP repressor binding site, overlaps with the TRP promoter sequence between bases -21 and +3. The core binding site is a palindrome of 18 BP. The TRP repressor binds tryptophan and can only bind to the operator when it is complexed with tryptophan. The repressor is a dimer of 2 subunits which have structural similarity to the CRP protein and LAC repressor. The repressor dimer has a structure with a central core and 2 flexible DNA reading heads each formed from the carboxyl terminal half of one subunit. Only when tryptophan is bound to the repressor are the reading heads the correct distance apart, and the side chains in the correct conformation, to interact with successive major grooves of the DNA at the TRP operator sequence. Tryptophan, the end product of the enzymes encoded by the TRP operon, therefore acts as a co-repressor and inhibits its own synthesis through end product inhibition. The repressor reduces transcription initiation by around 70-fold. This is a much smaller transcriptional effect than that mediated by the binding of the LAC repressor. Attenuator. At first, it was thought that the repressor was responsible for all of the transcriptional regulation of the TRP operon. However, it was observed that the deletion of a sequence between the operator and the TRP gene coding region resulted in an increase in both the basal and the activated repressed levels of transcription. This site is termed the attenuator and it lies towards the end of the transcribed leader sequence of 162 NT that precedes the TRP initiator codon. The attenuator is a row-independent terminator site which has a short GC-rich palindrome followed by 8 successive U residues. If this sequence is able to form a hairpin structure in the RNA transcript, then it acts as a highly strong terminator and only a 140 BP transcript is synthesized. The leader RNA structure. The leader sequence of the TRP operon RNA contains 4 regions of complementary sequence which can form different base paired RNA structures. These are termed sequences 1, 2, 3 and 4. The attenuator hairpin is the product of the base pairing of sequences 3 and 4, 3, 4 structure. Sequences in 2 are also complementary and can form a second 1, 2 hairpin. However, sequence 2 is also complementary to sequence 3. If sequences 2 and 3 form 2, 3 hairpin structure anti-termination, the 3, 4 attenuator hairpin cannot be formed and transcription termination will not occur. However, sequence 2 is also complementary to sequence 3. If sequences 2 and 3 form 2, 3 hairpin structure anti-termination, the 3, 4 attenuator hairpin cannot be formed and transcription termination will not occur. Under normal conditions, the formation of the 1, 2 and 3, 4 hairpins is energetically favorable. The leader peptide. The leader RNA sequence contains an efficient ribosome binding site and form a 14 amino acid leader peptide encoded by bases 27 to 68 of the leader RNA. The 10th and 11th codons of this leader peptide encode successive tryptophan residues, the end product of the synthetic enzymes of the TRP operon. This peptide has no obvious function as a polypeptide, and tryptophan is a rare amino acid. Therefore, the chances of 2 tryptophan codons in succession is low, and under conditions of low tryptophan availability, the ribosome would be expected to pause at this site. The function of this leader peptide is to determine tryptophan availability and to regulate transcription termination. Attenuation. Attenuation depends on the fact that transcription and translation are tight and are coupled in E. coli. Translation can occur as an mRNA is being transcribed. 3 feet end of the TRP leader peptide coding sequence overlaps complementary sequence 1. The 2 TRP codons are within sequence 1 and the stop codon is between sequences 1 and 2. The availability of tryptophan, the ultimate product of the enzyme synthesized by the TRP operon, is sensed through its being required in translation, and determines whether or not the terminator 3, 4 hairpin forms in the mRNA. As transcription of the TRP operon proceeds, the RNA polymerase pauses the end of sequence 2 until a ribosome begins to translate the leader peptide. Under conditions of high tryptophan availability, the ribosome rapidly incorporates tryptophan at the 2 TRP codons and thus translates to the end of the leader message. The ribosome is then occluding sequence 2N. As the polymerase reaches the terminator sequence, the 3, 4 hairpin can form, and transcription may be terminated. This is the process of attenuation. Alternatively, if tryptophan is in scarce supply, it will not be available as aminoacyl tRNA for translation, and the ribosome will then pause at the 2 TRP codons, occluding sequence 1. This leaves sequence 2 free to form a hairpin with sequence 3, known as the anti-terminator terminator 3, 4 hairpin cannot form, and transcription continues in direction of TRP genes and beyond. Thus the level of the end product, tryptophan, determines the probability that transcription will terminate early, attenuation rather than proceeding through the whole operon. The presence of tryptophan gives rise to AL0-fold repression of TRP operon transcription through the process of attenuation alone. Combined with contribution by the TRP repressor, 70-fold, this means that tryptophan levels exert a 700-fold regulatory effect on expression from the TRP operon. Attenuation occurs in at least 6 operons that encode enzymes concerned with amino acid biosynthesis. For example, the His operon has a leader which encodes a peptide with 7 successive histidine codons. Not all of these other operons have the same combination of regulatory controls that are found in the TRP operon. The His operon has no repressor operator regulation, and attenuation forms the only mechanism of feedback control. 1.3 Transcriptional regulation by alternative sigma factors. The alpha-2 BB W core enzyme of RNA polymerase is unable to start transcription at promoter sites. In order to specifically recognize the consensus -35 and -10 elements of general promoters, it requires a sigma factor subunit. This subunit is only required for transcription initiation, being released from the core enzyme after initiation and before RNA elongation takes place. Thus, sigma factors appear to be bifunctional proteins that simultaneously combine to core RNA polymerase and recognize specific promoter sequences in DNA. Many bacteria, including E. coli, produce a set of sigma factors that recognize different sets of promoters. Transcription initiation from single promoters or small groups of promoters is regulated commonly by single transcriptional repressors, such as the LAC repressor, or transcriptional activators, such as the CAMP receptor protein, CRP. However, some environmental conditions require a massive change in the overall pattern of gene expression in the cell. Under such circumstances, bacteria may use a different set of sigma factors to direct RNA polymerase binding to different promoter sequences. This process allows the diversion of the cell's basic transcription machinery to the specific transcription of different classes of genes. Promoter recognition. The binding of an alternative sigma factor to RNA polymerase can confer a new promoter specificity on the enzyme responsible for the general RNA synthesis of the cell. Comparisons of promoters activated by polymerase complex to specific sigma factors show that each sigma factor recognizes a different combination of sequences centered approximately around the -35 intersites. It seems likely that sigma factors themselves contact both of these regions, with the region J being most important. The sigma 70 subunit is the most common sigma factor in E. coli, which is responsible for recognition of general promoters which have consensus -35 and -10 elements. Heat shock. The response to heat shock is one example in E. coli where gene expression is altered significantly by the use of different sigma factors. When E. coli is subjected to an increase in temperature, the synthesis of a set of around 17 proteins, called heat shock proteins, is induced. If E. coli is transferred from 37 to 42 degrees Celsius, this burst of heat shock protein synthesis is transient. However, if the increase in temperature is more extreme, such as to 50 degrees Celsius, where growth of E. coli is not possible, then the heat shock proteins are the only proteins synthesized. The promoters for E. coli heat shock protein encoding genes are recognized by a unique form of RNA polymerase holoenzyme containing a variant sigma factor, sigma 32, which is encoded by the RPOH gene. Sigma 32 is a minor protein which is much less abundant than sigma 70. Holoenzyme containing sigma 32 acts exclusively on promoters of heat shock genes and does not recognize the general consensus promoters of most of the other genes Fig. Heat shock promoters accordingly have different sequences to other general promoters which bind to sigma 70. Sporulation of Bacillus subtilis. Sporulation in Bacillus subtilis. Vegetatively growing B subtilis. Sporulation in Bacillus subtilis. Vegetatively growing B subtilis cells form bacterial spores in response to a suboptimal environment. The formation of a spore or sporulation requires drastic changes in gene expression, including the cessation of the synthesis of almost all of the proteins required for vegetative existence as well as the production of proteins which are necessary for the resumption of protein synthesis when the spore germinates under more optimal conditions. The process of spore formation involves the asymmetrical division of the bacterial cell into two compartments, the phorospore, which forms the spore, and the mother cell, which is eventually discarded. This system is considered one of the most fundamental examples of cell differentiation. The RNA polymerase in B subtilis is functionally identical to that in E. coli. The vegetatively growing B subtilis contains a diverse set of sigma factors. Sporulation is regulated by a further set of sigma factors in addition to those of the vegetative cell. Different sigma factors are specifically active before cell partition occurs, in the phorospore and in the mother cell. Cross-regulation of this compartmentalization permits the phorospore and mother cell to tightly coordinate the differentiation process. Bacteriophage sigma factors. Some bacteriophages provide new sigma subunits to endow the host RNA polymerase with a different promoter specificity and hence to selectively express their own phage genes, e.g. phage T4 in E. coli and sp01 in B. subtilis. This strategy is an effective alternative to the need for the phage to encode its own complete polymerase, e.g. bacteriophage T7. The B. subtilis bacteriophage sp01 expresses a cascade of sigma factors in sequence to allow its own genes to be transcribed at specific stages during virus infection. Initially, early genes are expressed by the normal bacterial hello enzyme. Among these early genes is the gene encoding sigma 28, which then displaces the bacterial sigma factor from the RNA polymerase. The sigma 28 containing hello enzyme is then responsible for expression of the middle genes. The phage middle genes include genes 33 and 34 which specify a further sigma factor that is responsible for the specific transcription of late genes. In this way, the bacteriophage uses the host's RNA polymerase machinery and expresses its genes in a defined sequential order. D2. Regulation of gene expression in eukaryotes. 2. 1. Chromatin modifications, heterochromatization, and genome expression. Chromatin is the complex of genomic DNA and chromosomal proteins present in the eukaryotic nucleus. Chromatin structure is hierarchic, ranging from the two lowest levels of DNA packaging the nucleosome and the 30 nanometers chromatin fiber to the metaphase chromosomes, which represent the most compact form of chromatin in eukaryotes and occur only during nuclear division. After division, the chromosomes become less compact and cannot be distinguished as individual structures. When non-dividing nuclei are examined by light microscopy, all that can be seen is a mixture of lightly and darkly staining areas within the nucleus. The dark areas, which tend to be concentrated around the periphery of the nucleus, are called heterochromatin and contain DNA that is still in a relatively compact organization, although still less compact than in the metaphase structure. Two types of heterochromatin are recognized. 1. Constitutive heterochromatin is a permanent feature of all cells and represents DNA that contains no genes and so can always be retained in a compact organization. This fraction includes centromeric and telomeric DNA as well as certain regions of some other chromosomes. For example, most of the human Y chromosome is made of constitutive heterochromatin. 2. Facultative heterochromatin is not a permanent feature but is seen in some cells some of the time. Facultative heterochromatin is thought to contain genes that are inactive in some cells or at some periods of the cell cycle. When these genes are inactive, their DNA regions are compacted into heterochromatin. It is assumed that the organization of heterochromatin is so compact that proteins involved in gene expression simply cannot access the DNA. In contrast, the remaining regions of chromosomal DNA, the parts that contain active genes, are less compact and permit entry of the expression proteins. These regions are called euchromatin and they are dispersed throughout the nucleus. First, the degree of chromatin packaging displayed by a segment of a chromosome determines whether or not genes within that segment are expressed. Second, if a gene is accessible, then its transcription is influenced by the precise nature and positioning of the nucleosomes in the region where the transcription initiation complex will be assembled. 2.1.1 Regulation of gene expression by histone. Modifications. Histone proteins can undergo various types of modification, the best studied of these being histone acetylation, the attachment of acetyl groups to lysine amino acids in the N-terminal regions of each of the core molecules. These N-termini form tails that protrude from the nucleosome core octamer and their acetylation reduces the affinity of the histones for DNA and possibly also reduces the interaction between individual nucleosomes that leads to formation of the 30 nanometers chromatin fiber. The histones in heterochromatin are generally unacetylated whereas those in functional domains are acetylated, a clear indication that this type of modification is linked to DNA packaging. Histone acetyl transferases, HATs, the enzymes that add acetyl groups to histones. For example, one of the first HATs to be discovered, the tetrahymena protein called P55, which was known to activate assembly of the transcription initiation complex. One way in which silencing can be implemented is by removing acetyl groups from histone tails, and hence reversing the transcription activating effects of the HATs. This is the role of the histone deacetylases, HDACs. 2.2.2. Gene regulation by DNA methylation. Chromatin modification is not the only process that can bring about genome silencing. DNA methylation can also repress gene activity. DNA methylation can repress gene activity. Initially this was thought to be quite distinct from histone modification, but now we are beginning to see links between the two activities. In eukaryotes, cytosine bases in chromosomal DNA molecules are sometimes changed to 5-methylcytosine by the addition of methyl groups by enzymes called DNA methyltransferases. Cytosine methylation is relatively rare in lower eukaryotes, but in vertebrates up to 10% of the total number of cytosines in a genome is methylated, and in plants the figure can be as high as 30%. The methylation pattern is not random, instead being limited to the cytosine in some copies of the sequence is 5. CG3. And, in plants, 5. CNG3. Two types of methylation activity have been distinguished. The first is maintenance methylation which, following genome replication, is responsible for adding methyl groups to the newly synthesized strand of DNA at positions opposite methylated sites on the parent strand. The maintenance activity therefore ensures that the two daughter DNA molecules retain the methylation pattern of the parent molecule. The second activity is de novo methylation, which adds methyl groups at totally new positions and so changes the pattern of methylation in a localized region of the genome. It was originally thought that the first DNA methyltransferase, DNMT1, was responsible for both types of methylation in mammalian cells. It was subsequently discovered that knockout mice that have an inactivated gene for DNMT1 can still carry out de novo methylation. This led to the search for new enzymes and the eventual discovery of DNMT3 and DNMT3B, which are now considered to be the main de novo methylases of mammals, with DNMT1 primarily responsible for the maintenance activity. Methylation results in repression of gene activity. This has been shown by experiments in which methylated or unmethylated genes have been introduced into cells by cloning and their expression levels measured. Expression does not occur if the DNA sequence is methylated. The link with gene expression is also apparent when the methylation patterns in chromosomal DNAs are examined, these showing that active genes are located in unmethylated regions. For example, in humans, 40–50% of all genes are located close to CPG islands, with the methylation status of the CPG island reflecting the expression pattern of the adjacent gene. Housekeeping genes, those that are expressed in all tissues, have unmethylated CPG islands, whereas tissue-specific genes are unmethylated only in those tissues in which the adjacent gene is expressed. How methylation influences genome expression was a puzzle for many years. Now it is known that methyl CPG binding proteins, MECPs, are components of both the SYN3 and NERD histone deacetylase complexes. This discovery has led to a model in which methylated CPG islands are the target sites for attachment of HDAC complexes that modify the surrounding chromatin in order to silence the adjacent genes. Dosage compensation by X chromosome. Inactivation in mammals. It occurs because females have two X chromosomes whereas males have only one. If both of the female X chromosomes were active, then proteins coded by genes on the X chromosome might be synthesized at twice the rate in females compared with males. To avoid this undesirable state of affairs, one of the female X chromosomes is silenced and is seen in the nucleus as a condensed structure called the Barr body, which is comprised entirely of heterochromatin. Silencing occurs early in embryo development and is controlled by the X inactivation center, a discrete region present on each X chromosome. In a cell undergoing X inactivation, the inactivation center on one of the X chromosomes initiates the formation of heterochromatin, which spreads out from the nucleation point until the entire chromosome is affected, with the exception of a few short segments containing small clusters of genes that remain active. The process takes several days to complete. 2.2. Regulatory sequences and transacting factors. Cis and trans are two important terms relevant to the genetic study of gene regulation in bacteria and eukaryotes. A locus is cis-acting on a second locus if it must be on the same DNA molecule in order to have an effect. The operator is a cis-acting element because it works only when physically attached to the gene whose expression it regulates. A locus is transacting if it can affect a second locus even when on a different DNA molecule. The gene for the lactose repressor, LAC, is transacting because it can regulate expression of the lactose operon even when removed from the Escherichia coli chromosome and placed on a plasmid. To a molecular biologist, a cis-acting regulatory element is usually a target site for a DNA-binding protein, upstream of the gene whose expression is being regulated. A transacting element is the regulatory protein itself, which can diffuse through the cell from its site of synthesis to its DNA-binding site. 2.2.1 RNA polymerase II genes, promoters and enhancers, regulatory sequences. RNA polymerase II, RNA pol II, is located in the nucleoplasm. It is responsible for the transcription of all protein-coding genes and some small nuclear RNA genes. The pre-mRNAs must be processed after synthesis by cap formation at the 5' end of the RNA and poly A addition at the 3' end, as well as removal of introns by splicing. Promoters. Many eukaryotic promoters contain a sequence called the tau-tau box around 25 to 35 BP upstream from the start site of transcription Fig. 1. It has 7 BP consensus sequence 5' tau-tau AT-3, although it is now known that the protein which binds to the tau box, TBP, binds to an 8 BP sequence that includes an additional downstream base pair, whose identity is not important. The tau box acts in a similar way to an E. coli promoter -10 sequence to position the RNA poly for correct transcription initiation. While the sequence around the tau box is critical, the sequence between the tau box and the transcription start site is not critical. However, the spacing between the tau box and the start site is important. Around 50% of the time, the start site of transcription is an adenine residue. Some eukaryotic genes contain an initiator element instead of a tau box. The initiator element is located around the transcription start site. Many initiator elements have AC at position -1 and an A at +1. Other promoters have neither a tau box nor an initiator element. These genes are generally transcribed at low rates, and initiation of transcription may occur at different start sites over a length of up to 200 BP. These genes often contain a GC-rich 20 to 50 BP region within the first 1 to 200 BP upstream from the start site. Upstream regulatory elements. The low activity of basal promoters is greatly increased by the presence of other elements located upstream of the promoter. These elements are found in many I genes which vary widely in their levels of expression in different tissues. Two common examples are the SP1 box, which is found upstream of many genes both with and without tau boxes, and the CCAAT box. Promoters may have one or both or multiple copies of these sequences. These sequences, which are often located within 1 to 200 BP upstream from the promoter, are referred to as upstream regulatory elements, UREs, and play an important role in ensuring efficient transcription from the promoter. Enhancers. Transcription from many eukaryotic promoters can be stimulated by control elements that are located many thousands of base pairs away from the transcription start site. This was first observed in the genome of the DNA virus SV40. A sequence of around 100 BP from SV40 DNA can significantly increase transcription from a basal promoter even when it is placed far upstream. Enhancer sequences are characteristically 10 to 200 BP long and contain multiple sequence elements which contribute to the total activity of the enhancer. They may be ubiquitous or cell type specific. Classically, enhancers have the following general characteristics. They exert strong activation of transcription of a linked gene from the correct start site. They activate transcription when placed in either orientation with respect to linked genes. They are able to function over long distances of more than 1 kilobit, whether from an upstream or downstream position relative to the start site. They exert preferential stimulation of the closest of two tandem promoters. However, as more enhancers and promoters have been identified, it has been shown that the upstream promoter and enhancer motifs overlap physically and functionally. There seems to be a continuum between classic enhancer elements and those promoter elements which are orientation specific and must be placed close to the promoter to have an effect on transcriptional activity. 2.2.2. Regulation of transcription initiation. Transcription initiation is the stage at which the critical controls over the expression of individual genes, i.e. those controls that have greatest impact on the biochemical properties of a cell, are exerted. This is perfectly understandable. It makes sense that transcription initiation, being the first step in genome expression, should be the stage at which primary regulation occurs, this being the level of regulation that determines which genes are expressed. We looked at how chromatin structure can influence gene expression by controlling the accessibility of promoter sequences to RNA polymerase and its associated proteins. This is just one way in which initiation of transcription can be regulated. With bacteria, it is possible to make a clear distinction between constitutive and regulatory forms of control over transcription initiation. In bacteria, the RNA polymerase has a strong affinity for its promoter and the basal rate of transcription initiation is relatively high for all but the weakest promoters. With most eukaryotic genes, the reverse is true. The RNA polymerase II and III pre-initiation complexes do not assemble efficiently and the basal rate of transcription initiation is therefore very low, regardless of how strong the promoter is. In order to achieve effective initiation, formation of the complex must be activated by additional proteins. Some of these could be defined as constitutive activators, in that they work on many different genes and seem not to respond to any external signals. Others could be termed regulatory activators because they target a limited number of genes and do respond to external signals. 2.2.3 Constitutive transcription factor SP1. SP1 binds to a GC-rich sequence with the consensus sequence GGGCGG. It is a constitutive transcription factor whose binding site is found in the promoter of many housekeeping genes. SPI is present in all cell types. It contains 3 zinc finger motifs and has been shown to contain 2 glutamine-rich transactivation domains. The glutamine-rich domains of SPI have been shown to interact specifically with TAF2-110, one of the TAF2s which bind to the tau-tau binding protein TBP, to make up TFIID. This represents one target through which SBI may interact with and regulate the basal transcription complex. 2.2.4 Activators of eukaryotic transcription initiation. Any protein that stimulates transcription initiation is called an activator. Initially, it was imagined that all activators were sequence-specific DNA-binding proteins, some recognizing upstream promoter elements and influencing transcription initiation only at the promoter to which these elements are attached, and others targeting sites within enhancers and influencing transcription of several genes at once. As with bacteria, eukaryotic enhancers can be some distance from their genes. Their target specificity is ensured by the presence of insulators at either side of each functional domain, preventing the enhancers within that domain from influencing gene expression and adjacent domains. Whether bound to an upstream promoter element or to a more distant enhancer, the activator, according to the traditional view, stabilizes the pre-initiation complex by making contact with it. This traditional view still holds for the majority of activators that have been identified but cannot be looked upon as all-encompassing. We have already seen that some proteins that were initially identified as activators are now recognized as components of chromatin modification complexes such as SAGA and SWI/SNF. Other proteins classed as activators influence gene expression by introducing bends and other distortions into DNA, possibly as a prelude to chromatin modification, or possibly to bring together proteins attached to non-adjacent sites, enabling the bound factors to work together in a structure that has been called an enhanceosome. An example of an activator that works in this way is SRY, which is the primary protein responsible for determining sex in mammals. Still, other activators have no DNA binding properties and they stimulate transcription simply by forming protein-protein contacts with the pre-initiation complex. Activators have been looked upon as important in initiation by RNA polymerases 2 and 3, but their role at RNA polymerase I promoters has been less well defined. RNA polymerase I is unusual in that it transcribes just a single set of genes, the multiple copies of the transcription unit containing the 28S, 5.8S and 18S rRNA sequences. These genes are expressed continuously in most cells, but the rate of transcription varies during the cell cycle and is subject to a certain amount of tissue-specific regulation. The regulatory mechanism has not been described in detail, but recent research has suggested a role for the RNA polymerase I termination factor. This factor, called TTF1 in mice and REB1P in Saccharomyces cerevisiae, was first identified as an activator of RNA polymerase II transcription. It appears that the termination factor may also activate RNA polymerase I transcription, a binding site for it having been located immediately upstream of the promoter for the rRNA transcription unit. Contacts between activators and the pre-initiation complex, a critical feature of the traditional type of activator, those that bind to upstream promoter elements or to enhancers, is a contact that is formed with the pre-initiation complex. The part of the activator that makes this contact is called the activation domain. Structural studies have shown that although activation domains are variable, most of them fall into one of three categories. Acidic domains are ones that are relatively rich in acidic amino acids, aspartic acid and glutamic acid. This is the commonest category of activation domain. Glutamine-rich domains are often found in activators whose DNA binding motifs are of the homeodomain or PRU type. Proline-rich domains are less common. A number of protein-protein interaction studies have suggested that direct contacts could be made between different activators in various parts of the complex, with TBP, various TAFs, TFIIB, TFIIH and RNA polymerase II all implicated as partners in different interactions. An alternative possibility was raised when a large protein complex called the mediator was identified in yeast. The mediator forms a physical contact between activators in the C-terminal domain of RNA polymerase II, suggesting that rather than direct interaction between an activator and the pre-initiation complex, the signal is transduced by the mediator. This hypothesis was strengthened when it was shown that the mediator possesses a protein kinase activity that enables it to phosphorylate the CTD of RNA polymerase II, stimulating promoter clearance. Current opinion tends to the view that a mediator is an obligatory component of the RNA polymerase II pre-initiation complex, and that the stimulatory effects of all activators pass through the mediator. The possibility that some activators bypass the mediator and have a direct effect on one or other part of the pre-initiation complex cannot, however, be discounted. 2.2.5 Repressors of eukaryotic transcription initiation. Most of the research on regulation of transcription initiation in eukaryotes has concentrated on activation, partly because the low level of basal initiation occurring at RNA polymerase II and 3 promoter suggests that the repression of initiation, which is so important in bacteria, is unlikely to play a major part in control of eukaryotic transcription. This view is probably incorrect because a growing number of DNA binding proteins that repress transcription initiation are being discovered, these proteins binding to upstream promoter elements or to more distant sites in silencers. Some influence genome expression in a general way through histone dysylation or DNA methylation, but others have more specific effects at individual promoters. The yeast repressors called MO1 and NC2, for example, inhibit assembly of the pre-initiation complex by binding directly to TBP and disrupting its activity. MO1 causes TBP to dissociate from the DNA, and NC2 prevents further assembly of the complex on the bound TBP. Another indication of the importance of repression in eukaryotic transcription comes from the demonstration that some proteins can exert both activating and repressing effects, depending on the circumstances. NC2, for example, represses initiation of transcription from promoters with a TATA box but has an activating effect on promoters that lack the TATA sequence. Relatively little is known about the precise interactions occurring between repressors and the pre-initiation complex. A variety of inhibition domains, the converse of an activation domain, have been identified in eukaryotic repressors, several of which are rich in prolines, but no general patterns have emerged. The direct interactions with TBP displayed by MO1 and NC2 argue against the involvement in repression of a complex equivalent to the mediator that is required for gene activation. 2.2.6 Hormonal regulation, steroid hormone receptor. Many transcription factors are activated by hormones which are secreted by one cell type and transmit a signal to a different cell type. One class of hormones, the steroid hormones, are lipid-soluble and can diffuse through cell membranes to interact with transcription factors called steroid hormone receptors. In the absence of the steroid hormone, the receptor is bound to an inhibitor and located in the cytoplasm Fig. 1. The steroid hormone binds to the receptor and releases the receptor from the inhibitor, allowing the receptor to dimerize and translocate to the nucleus. The DNA binding domain of the steroid hormone receptor then interacts with its specific DNA binding sequence, or response element, and this gives rise to activation of the target gene. Important classes of related receptors include the glucocorticoid, estrogen, retinoic acid and thyroid hormone receptors. The general model described above is not true for all of these. For example, the thyroid hormone receptors act as DNA-bound repressors in the absence of hormone. In the presence of the hormone, the receptor is converted from a transcriptional repressor to a transcriptional activator. 2.2.7 Regulation by phosphorylation, staph proteins. Many hormones do not diffuse into the cell. Instead, they bind to cell surface receptors and pass a signal to proteins within the cell through a process called signal transduction. This process often involves protein phosphorylation. Interferon gamma induces phosphorylation of a transcription factor called STAT-alpha through activation of the intracellular kinase called Janus-activated kinase JAK. When STAT1-alpha protein is unphosphorylated, it exists as a monomer in the cell cytoplasm and has no transcriptional activity. However, when STAT1-alpha becomes phosphorylated at a specific tyrosine residue, it is able to form a homodimer which moves from the cytoplasm into the nucleus. In the nucleus, STAT1-alpha is able to activate the expression of target genes whose promoter regions contain a consensus DNA binding. 2.2.8 Embryonic development. The homeobox is a conserved DNA sequence which encodes a helix-turn-helix DNA binding protein structure called the homeodomain. The homeodomain was first discovered in the transcription factors encoded by homeotic genes of Drosophila. Homeotic genes are responsible for the correct specification of body parts. For example, mutation of one of these genes, antennapedia, causes the flight of formal leg where the antenna should be. These genes are very important in spatial pattern formation in the embryo. The homeobox sequence has been conserved between a wide range of eukaryotes, and homeobox-containing genes have been shown to be important in mammalian development. In Drosophila and mammals, the homeobox genes are arranged in gene clusters in which homologous genes are in the same order. The gene homologs are also expressed in a similar order in the embryo on the anterior to posterior axis. This suggests that the conserved homeobox-encoded DNA binding domain is characteristic of transcription factors which have a conserved function in embryonic development. 2.2.9 Translational control. Because of the different natures of the mRNA in prokaryotes and eukaryotes, i.e. polycistronic versus monocistronic, and the absence of the nuclear membrane in the former, different possibilities exist for the control of translation. In prokaryotes, the structure formed by regions of the mRNA can obscure ribosome binding sites, thus reducing translation of some cistrons relative to others. The formation of stems and loops can inhibit exonucleases and give certain regions of the polycistronic mRNA a greater half-life, and hence a greater chance of translation than others. Several operons encoding ribosomal proteins show an interesting form of translational control in E. coli where a region of the mRNA has a tertiary structure that resembles the binding site for a ribosomal protein encoded by the mRNA. If there is insufficient rRNA available for the translation product to bind to, it will bind to its own mRNA and prevent further translation. Prokaryotes sometimes make short antisense RNA molecules that form duplexes near the ribosome binding site of certain mRNAs, thus inhibiting translation. Eukaryotes generally control the amount of specific proteins by varying the level of transcription of the gene and/or by RNA processing, but some controls occur in the cytoplasm. The presence of multiple copies of 5' AUUUA3, usually in the 3' non-coding region, marks the mRNA for rapid degradation and thus limited translation. Another form of translational control involves proteins binding directly to the mRNA and preventing translation. This RNA is called masked mRNA. In appropriate circumstances, the mRNA can be translated when the protein dissociates. Some non-coding sequences can cause mRNA to be located in a specific part of the cytoplasm and, when translated, can give rise to a gradient of protein concentration across the cell. Proteins, bacteriophage and viral transcripts in many mRNAs for hormones in eukaryotes, e.g. pro-opiomelanocortin, are translated to give a single polypeptide chain that is cleaved subsequently by specific proteases to produce multiple mature proteins from one translation product. The parent polypeptide is called a polyprotein. Protein targeting. It has been discovered that the ultimate cellular location of proteins is often determined by specific, relatively short, amino acid sequences within the proteins themselves. These sequences can be responsible for proteins being secreted, imported into the nucleus, or targeted to other organelles. The greater complexity of a eukaryotic cell means that there are more types of targeting in eukaryotes. Protein secretion in both prokaryotes and eukaryotes involves a signal sequence in the nascent protein, and specific proteins are, in the latter, an RNP particle, signal recognition particle, SERP, that recognizes it Fig.1. If a cytosolic ribosome begins to translate an mRNA encoding a protein that is to be secreted, SERP binds to the ribosome and the emerging polypeptide and arrests translation. SERP is capable of recognizing ribosomes with a nascent chain containing a signal sequence, signal peptide, which is composed of about 13 to 36 amino acids having at least one positively charged residue followed by a hydrophobic core of 10 to 15 residues followed by a small, neutral residue, often ALA. SERP with the arrested ribosome binds to a receptor, SERP receptor or docking protein, on the cytosolic side of the endoplasmic reticulum, and, when the ribosome becomes attached to ribosome receptor proteins on the ER, SERP is released and can be reused. The ribosome is able to continue translation, and the nascent polypeptide chain is pushed through into the lumen of the ER. As it passes through, signal peptidase cleaves off the signal peptide. When the protein is released into the ER, it is usually modified, often by glycosylation, and different patterns of glycosylation seem to control the final location of the protein. Other peptide sequences in proteins are responsible for their cellular location. Different N-terminal sequences can cause proteins to be imported into mitochondria or chloroplasts, and the internal sequence Li-Li-Arg-Li, or any 5 consecutive positive amino acids, can be a nuclear localization signal NLS, causing the protein containing it, e.g. histone, to be imported into the nucleus. Protein modification. A newly translated polypeptide does not always immediately generate a functional protein. Apart from correct folding and the possible formation of disulfide bonds, there are a number of other alterations that may be required for activity. These include cleavage and various covalent modifications. Cleavage is very common, especially trimming by amino and carboxypeptidases, but the removal of internal peptides also occurs, as in the case of insulin. Signal sequences are also usually cleaved off secreted proteins, and, where proteins are made as parts of polyproteins, they must be cleaved to release the component proteins. Ubiquitin is made as a polyprotein containing multiple copies linked end to end, and this must be cleaved to generate the individual ubiquitin molecules. Chemical modifications are many and varied and have been shown to take place on the N and C termini, as well as on most of the 20 amino acid side chains, with the exception of ALA, GLY, LI, LU, MET and VAL. The modifications include acetylation, hydroxylation, phosphorylation, methylation, glycosylation and even the addition of nucleotides. Hydroxylation of PRO is common in collagen, and some of the histone proteins are often acetylated. The activity of many enzymes, such as glycogen phosphorylase and some transcription factors, is controlled by phosphorylation. Protein degradation. Different proteins have very different half-lives. Regulatory proteins tend to turn over rapidly and cells must be able to dispose of faulty and damaged proteins. In eukaryotes, it has been discovered that the N-terminal residue plays a critical role in inherent stability. 8 N-terminal amino acids ALA, CYs, GLY, MET, PRO, ESER, THR, VAL correlate with stability T1/2 greater than 20 hours 8. ARG, HIS, LI, LU, LI, Phi, TRP, TR with short T1/2, 2-30 minutes, and 4 ASN, ASP, GIN, GLU are destabilizing following chemical modification. A protein that is damaged, modified or has an inherently destabilizing N-terminal residue becomes ubiquitinated by the covalent linkage of molecules of the small, highly conserved protein, ubiquitin, via its C-terminal BLY, to lysine residues in the protein. The ubiquitinated protein is digested by a 26S protease complex proteasome in a reaction that requires ATP and releases intact ubiquitin for reuse. D3. Eukaryotic transcription factors. Transcription factors other than the general transcription factors of the basal transcription complex were first identified through their affinity for specific motifs in promoters, upstream regulatory elements, UREs, or enhancer regions. These factors have two distinct activities. Firstly, they bind specifically to their DNA binding site and, secondly, they activate transcription. These activities can be assigned to separate protein domains called activation domains and DNA binding domains. In addition, many transcription factors occur as homo or heterodimers, held together by dimerization domains. A few transcription factors have ligand binding domains which allow regulation of transcription factor activity by binding of an accessory small molecule. The steroid hormone receptors are an example containing all four of these types of domain. Mutagenesis of the yeast transcription factors GAL4 and GCN4 showed that their DNA binding and transcription activation domains were in separate parts of the proteins. Experimentally, these activation domains were fused to the bacterial LEXA repressor. These hybrid fusion proteins activated transcription from a promoter containing the LEXA operator sequence, indicating that the transcriptional activation function of the yeast proteins was separable from their DNA binding activity. These type of experiments are called domain swap experiments. 3.1 The helix-turn-helix domain. This domain is characteristic of DNA binding proteins containing a 60 amino acid homeodomain which is encoded by a sequence called the homeobox. In the Antennopediae transcription factor of Drosophila, this domain consists of 4 alpha helices in which helices 2 and 3 are at right angles to each other and are separated by a characteristic beta tum. The characteristic helix-turn-helix structure, Fig.1, is also found in bacteriophage DNA binding proteins such as the phagic CRO repressor, LAC and TRP repressors, and CAMP receptor protein, CRP. The domain binds so that one helix, known as the recognition helix, lies partly in the major groove and interacts with the DNA. The recognition helices of 2 homeodomain factors bicoid and Antennopedia can be exchanged, and this swaps their DNA binding specificities. Indeed, the specificity of this interaction is demonstrated by the observation that the exchange of only one amino acid residue swaps the DNA specificities. 3.2 The zinc finger domain. This domain exists in two forms. The C2H2 zinc finger has a loop of 12 amino acids anchored by 2 cysteine and 2 histidine residues that tetrahedrally coordinate a zinc ion, Fig.2. This motif folds into a compact structure comprising 2 beta strands and 1 alpha helix, the latter binding in the major groove of DNA, Figure 2b. The ahelical region contains conserved basic amino acids which are responsible for interacting with the DNA. This structure is repeated 9 times in TFIIIA, the RNA pol 3 transcription factor. It is also present in transcription factor SPI, 3 copies. Usually, 3 or more C2H2 zinc fingers are required for DNA binding. A related motif, in which the zinc ion is coordinated by 4 cysteine residues, occurs in over 100 steroid hormone receptor transcription factors. These factors consist of homo or heterodimers, in which each monomer contains 2 C4 zinc finger, motifs Fig. The two motifs are now known to fold together into a more complex conformation stabilized by zinc, which binds to DNA by the insertion of 1A helix from each monomer into successive major grooves, in a manner reminiscent of the helix-turn-helix proteins. The basic domain. A basic domain is found in a number of DNA binding proteins and is generally associated with one or other of 2 dimerization domains, the leucine zipper or the helix loop helix HLH motif. These are referred to as basic leucine zipper BZIP or basic HLH proteins. Dimerization of the proteins brings together 2 basic domains which can then interact with DNA. 3.3. Leucine zippers. Leucine zipper proteins contain a hydrophobic leucine residue at every 7th position in a region that is often at the C-terminal part of the DNA binding domain. These leucines lie in an alpha helical region and the regular repeat of these residues forms a hydrophobic surface on one side of the alpha helix with a leucine every second turn of the helix. These leucines are responsible for dimerization through interactions between the hydrophobic faces of the alpha helices, see Fig. This interaction forms a coiled-coil structure. BZIP transcription factor contain a basic DNA binding domain N-terminal to the leucine zipper. This is present on an alpha helix which is a continuation from a leucine zipper alpha helical C-terminal domain. The N-terminal basic domains of each helix form a symmetrical structure in which each basic domain lies along the DNA in opposite directions, interacting with a symmetrical DNA recognition site so that the protein in effect forms a clamp around the DNA. The leucine zipper is also used as a dimerization domain in proteins that use DNA binding domains other than the basic domain, including some homeodomain proteins. The helix loop helix domain. The overall structure of this domain is similar to the leucine zipper, except that a non-helical loop of polypeptide chain separates 2 alpha helices in each monomeric protein. Hydrophobic residues on one side of the C-terminal helix allow dimerization. This structure is found in the MyOD family of